So our next presentation is going to be on snubfin and humpback dolphins. Again, there's a bit of a team effort on working this project up. It has a few different components to it, looking at abundance, acoustic monitoring, and also genetic connectivity. And Josh Smith from Murdoch University will lead off, and joined by Chandra Salgado from Curtin University.
Okay, good morning, everyone. So this was a relatively large collaborative project that involved all those people up there, as well as a number of traditional owners and ranger groups, which I'll be able to identify a little bit into the talk. A little caveat is that I'm going to be presenting the relative abundance and genetic data. That's not really my forte. That's mainly Alex and Lars who are sort of doing that, but they couldn't be here, so I'm going to be presenting that. And then that's going to be followed by Chandra, who will be presenting the passive acoustic monitoring data. Okay, so both of these species, humpback dolphins and snubfin dolphins, are endemic to Australia, and they pretty much occur in small populations that are fragmented coastal populations. And that's pretty much the crux of the results that we found from this Kimberley surveys, is consistent with what we've been finding in Western Australia and other parts within Queensland, is this characteristic here. So what that means is that there's a high level of interaction with human coastal activities. With some of these studies, particularly in Queensland, where there's a bit more information, I guess, they're finding limited genetic connectivity in distances of less than about 350 kilometres. And so with legislation, what we have here is a difference between IUCN listed as near threatened, but within Australia, Commonwealth and state legislation has them as migratory cetacean. That's pretty much because there's just not enough information to warrant a lower level of classification than that, which kind of bears migratory potentially is not the best classification for these species of dolphins. So, given that there is data deficiency, this is particularly the case within the Kimberley. So that brings me to the aims of the project, which was essentially to assess the relative abundance and the genetic connectivity of a couple of different populations of these species within the Kimberley. And that's particularly within these three areas that are circled, Roebuck Bay, Cone Bay, and Cambridge Gulf. And then there was another component, which was applying the feasibility of passive acoustic methods for monitoring these populations, which is potentially a slightly more cost-effective approach than visual surveys. Now, as I said, generally we don't have enough information of these species within the Kimberley, and so an integral component of this project was working with traditional owner ranger groups in order to coordinate with them, uptake the information they had on the occurrence of these species in particular areas so that we could do strategic sampling, and then also work with them in the field, and particularly that included the Dampier-Mangari, Balangara, the Yaru, just to mention a few amongst others. Okay, so in relation to the methods, relative abundance, essentially this relied on boat-based photo identification, taking photographs of the dorsal fins of the dolphins so that we could individually identify them and determine encounter rates and relative abundance. This was following predetermined transit routes along the boats. The genetic connectivity using biopsy sampling, Paxarm rifles, and then the molecular analyses in the laboratory using mitochondrial DNA and microsatellite data to look at gene flow and genetic connectivity. And then essentially the efficacy of the PAM is looking at comparing the acoustic detections of these dolphins with visual detections from the boat-based surveys in the same spot. Okay, so with the boat-based surveys, they followed a structured sampling procedure, and by doing that means you can consistently apply the results in different sites within the Kimberley so that you can have a relative comparison. Here we've got the red boxed sites that was done within the WAMSI project, and the nice thing about this is that this coordinated and fed into other larger sampling programs that were done outside of WAMSI. And that's what we're seeing here with Roebuck Bay and Cygnet Bay, to give you a context in relation to the encounter rates and the numbers of dolphins that you would find in the areas that we did sample within the Kimberley, Cone Bay and Cambridge Gulf. As you can see, considerably fewer animals were sighted here, particularly in the Cambridge Gulf, than areas such as Roebuck Bay with high encounter rates. And this figure essentially, this is just looking at Combe Bay. Unfortunately, with Cambridge Gulf, sorry, there was really not enough interactions or encounters with the dolphins to develop a reliable photo identification discovery curve. This is looking at Combe Bay over the 2 years, 2014, 2015. Essentially, it's the cumulative index of the number of individually identified dolphins. And what we have here is the purple humpback dolphins, and the orange is the snubfin dolphins. And so, again, we're seeing with the humpback dolphins, it's plateauing at around about 20 individuals. That means we've seen pretty much all the same number of individuals that are going to be using that area. We're not seeing any newer ones. So we're looking at about 20 individuals there. With snubfin dolphins, it didn't quite plateau, but still would be expecting probably for that to remain around about 30 individuals. So I guess what that's showing here is these WAMSI-based, WAMSI funding surveys definitely demonstrate the notion that these species are occurring in low abundance. So conclusions in relation to the relative abundance, specifically it highlights the importance of Roebuck Bay, demonstrated by the high encounter rates, which is much higher than some of the other areas like particularly Admiralty Gulf and Combe Bay. So whilst Combe Bay does seem to support small local populations, it's probably best to identify them as communities, in that Combe Bay may not actually represent the entire home range for the snubfin humpback dolphins. What we did find, particularly looking at the Cambridge Gulf, is that photo ID isn't necessarily always a suitable technique. This was particularly the case in Cambridge Gulf, where there's very little vessel traffic. What we did find was that animals were more approachable when there was a lot more vessels around. So that has an impact on sort of sampling protocols that you end up using. And this was the case with biopsy sampling as well. And particularly what was evident with this project, given that they occur in small population sizes and relatively fragmented, isolated populations, is that in order to get any of this kind of information, the collaboration with the regional wildlife managers and the traditional owners and local stakeholders was essential to particularly get, in some cases outside of Combe Bay and Cambridge Gulf, to get opportunistic sightings on other trips that actually occurred. So that was essential. Future recommendations in relation to this is potentially targeted research on populations of management concerns. So it's unlikely that we're going to get abundance estimates or really good distribution data for these species over the entire Western Australia, and particularly the entire range over Australia. So it's probably going to make more sense to target research on populations that are currently or having projected exposures to threatening processes. If it's possible, it would be nice to select suitable sites, though, that are cost effective if we're trying to gain a handle on long-term population trends for these species, to keep a little bit of a baseline information. Okay, so that's the relative abundance aspect. So moving on to the genetic sampling. Don't worry too much about these pie charts. Essentially, that's mitochondrial DNA, but really just looking at gene flow. This is for the snubfin dolphins. The main point that came out of this was that there was very significant genetic differentiation between Roebuck Bay and the two other sites of Cygnet Bay and Combe Bay. And so what that suggests from a management implication is it's probably more sensible to manage Roebuck Bay as a separate population. There was a little bit of genetic differentiation between Cygnet Bay and Combe Bay, but or potential structure, I guess, within these sites. But unfortunately, limited samples means that the inference from this is kind of, we're not able to definitively say much about that yet until more samples are collected. And humpback dolphins, probably not unsurprisingly, but the main thing that came out of this is that there was very large genetic differentiation between the Pilbara and the Kimberley. So these are very separate populations. But given the distance between these two areas, but potentially not unsurprising. And again, with the humpback dolphins, there was a degree of structure and differentiation among the Kimberley sites, but unless there's more samples there, our inference is kind of hamstringed. So future recommendations for the genetic side of things, basically because there are limited sample sizes and because it is so logistically difficult to sample and very expensive to sample, it really requires considerable effort on integration with marine parks and ranger patrols, and they're going to be very important if we're going to get information in these kind of remote areas. And the use of, particularly for the genetics and so forth, the use of opportunistic surveys or opportunistic collection of data is still extremely important. From a strategic broad-scale sampling focus, really, given the genetic differentiation, we need to be looking at Eastern Kimberley and the Northern Territory in order to get samples from there so that we can understand the broader scale management units and the implications for the genetic differentiation over that much broader scale. So a lot's been done in the Kimberley, in the Western Kimberley now, but future efforts should probably focus in the Northern Territory and Eastern Kimberley. Potentially there's going to be more advanced techniques as well that we could delve into the samples that we have now, but time will tell with that. Okay, so moving on to passive acoustic monitoring, Chandra.
Okay, thank you very much, and thanks for the background, Josh. That makes my life much easier. All right. Okay, so Josh described the passive acoustic monitoring very briefly. So we looked at a range of sites to look at how effective it might be as an approach that actually allows a little bit more of a cost-effective way of monitoring dolphins. So this aspect was done by the two universities as a collaboration, Curtin University and Murdoch, and the passive acoustic work was mainly done by Josh for the acoustic. Curtin team did the first two, so we looked at describing the soundscape. So you need to understand the background noise levels and what's naturally in that environment to be able to assess passive acoustics as a tool. And then the Curtin team also looked at correlating the acoustics and the visual observations of dolphins, and Murdoch looked at the latter, basically objectives 3 and 4, which is identifying acoustic repertoire and behavioral context and assessing geographic variation. So I will show a spectrogram a little bit later, but just to orient those that aren't as aware of spectrograms, if we look at this, this figure, oh, sorry about that, just try that again. Huh. So, let me tell you. So that, I will continue just not to, not to lose time. So you did see that spectrogram. Basically, you have time on the x-axis and you've got frequency on the vertical. And so if you look across time, you can see what sounds you're actually seeing.
No star. It's the laser pointer to start.
Excellent. Okay. So this is just an example of one of those spectrograms. Okay, so there are several tools that were used. One was an acoustic logger that's bottom mounted, which is this one. This one's a highly calibrated system built at Curtin University at CMST, and it stays in the water over long periods. Sound traps were also used, so these are fantastic because they're quite lightweight. They also have a good capacity. And then finally, these recorders were used where you literally put a hydrophone in the water and you're able to basically target particular animals that you may be observing from a vessel. Okay, so this is the magic spectrogram. So what we did is first try to understand that soundscape. So it basically involved understanding what information we're looking at. So within spectrograms, obviously, you see lots of different sources of underwater noise. We had many different fish courses, so you can in fact use it to quantify fish courses or obtain relative abundance of vocalizing animals. You can also detect your vessel, so you get an idea of anthropogenic noise within the environment. You can pull out sounds from dolphins, and of course you have this energy up here, which is snapping shrimp. So we basically did this in 2 locations, Cygnet Bay and Roebuck Bay over the dry season. And what we found was that basically both locations are relatively pristine. So they were quite good places to actually test the passive acoustic monitoring. They were predominantly biological, and there were basically had low levels of anthropogenic noise, but of course when it did occur, it was mainly from vessels. And it was quite intense when it did occur. It occurred more often also during, for example, school holidays. What we found is that most of that noise was under the frequencies that dolphins mainly use, and so therefore we were able to effectively detect dolphins more frequently than, for instance, if we're in a very busy environment, such as actually the Swan River. Okay, so looking a bit closer, if we used Roebuck Bay as an example, which is an area that we focused on quite a bit, to look at PAM and its effectiveness. In this area, we ran boat surveys in the broader region, but we also did a higher effort in this small area that's frequently used by snubfin dolphins. That area, we placed one of those bottom-mounted loggers. And then we were able to basically compare what we saw in that area. And so this is basically the numbers of groups. Many are overlapping on each other with the size of the circle indicating group size. So we mainly saw within that snubfin dolphins, but we did see some of the other species. We saw overall the 111 groups. And from that, we actually were able to cross-compare to what we heard from the acoustic logger. So what we did here were the 3 different sources of acoustic signals from dolphins. The most common out of those, so of course you have whistles for communication that dolphins use, but you also have their biosonar, so echolocation clicks. And the echolocation clicks were in fact the ones that dominated. We're able to describe new whistle types for humpback dolphins. And when we cross-compared the surveys, we found that 67% of surveys with snubfin dolphin sighted had acoustic detections as well. So that's almost 70%. And what we found is the ones that we weren't detecting were mainly groups of single animals. So if we only looked at the ones that did not have single animals, there were multiple animals in the group, we detected over 90% of those using acoustics. So in conclusion, passive acoustic monitoring was in fact quite effective for detecting presence and absence of groups that were larger than one individual. Acoustics offers that really high temporal scale sampling. We were able to as well include within that larger catalog of sounds for humpback dolphins a number of whistles that then increases our capability for identifying individuals when using passive acoustic monitoring. What we still need to do is obviously be able to translate numbers into abundance, and that's still work to be done into the future. So what we can do in the future is look at ongoing monitoring using a passive acoustics. That will be indicative of presence, basically occupancy of areas, for instance, in Roebuck Bay. If we used an acoustic grid, we'd get that larger spatial scale of monitoring as well. So these basically are packages that can be put in and you leave them over months and then you pull them out. So that's sort of the advantage. You're not out daily doing putting in and pulling out. What needs to be done as well, and there's a wealth of data that's come out of it, is to look at habitat use and how that changes with a changing environment within days and over seasons. So looking at all the different environmental conditions that may predict the occupancy and use of an area like Roebuck Bay by dolphins and model that. And on top of that, of course, is look at the changing environment in terms of our use and how that may change their behavior and their use of the area. So there are changes occurring, for example, in Roebuck Bay, including new tourist operators. And so we don't yet know what that impact could potentially be or whether there is an impact or not. What we also need to do is provide this tool and training to local managers. So we had a fantastic opportunity to have the Yara on our boats, the country managers, and help us while we're out there. So we had lots of really great days and long hours on the boat together. This will help us understand things that we don't yet know, and then we can pass information back as well. And then finally, by developing automated detection algorithms, this will actually increase the efficiency and decrease the time spent and the resources used to use passive acoustic monitoring. Okay, a lot of people made this project happen. And in particular, we absolutely enjoyed the support from Yarra, the Badi Jawi, Danba Mangari Rangers, Environs Kimberley, Department of Parks and Wildlife, Broome Port Authority, and Clipper Pearls, and Marine Produce Australia. Thank you.